biologist. We are going to talk about mutations. What is a mutation? A mutation is a change in the nucleotide sequence of DNA. How are they caused? Many different ways, but some are environmental, some are inherited. There are lots of different ways that you can affect your DNA. We'll talk about that in detail on the next slide. And then, is it a bad thing? In general, not necessarily. So we think of mutation as maybe being something that leads to cancer, and that can be true. Most cancer cells have a mutation of some sort, especially in those tumor suppressor genes that we've discussed. But we also have mutations that have no effect or possibly a beneficial effect, or that might not really make a difference at all. I have two flower mutations being shown for you. This one is a little bit of albinism in your cute daisy here. Or sorry, this is a cosmo. The other ones are daisies. And so this one petal is not producing pigment that's caused by an albinism mutation. And then here we have one of my favorite mutations. This is called fascination. Fascination causes inflorescences and sometimes fruit created from a flower, which we call an inflorescence, to elongate. And so you get this extra long and wide daisy that has more reproductive parts than it ought to. And so that mutation may or may not be a bad thing for that flower. If it still gets fertilized and reproduces, then your flower probably doesn't care. So what causes mutations? The answer is a mutagen. So a mutagen is something that causes mutations, and a mutation being a change in the DNA. We can cause mutations via radiation, like X-rays or UV exposure. UV exposure specifically can cause skin cancer. The way that works is if I had a sequence of DNA that had a bunch of thymines together, which is relatively common. So A, T, T, G, C. I'm just making this up, but you get it. The complementary base pair below it would have a bunch of adenines to pair to those thymines above it, right? So I'm complementary base pairing these nucleotides to what's above it. UV radiation will come and hit these thymines, and it creates what we call thymine dimers. In thymine dimers, you create thymines that get a little bit too attracted to each other. And so these two thymines here will bend toward each other, and they will bond to each other instead of their A's. And then when this DNA is replicated, it sees that as just one single thymine because they're so closely bound together. And so it'll skip it. And now I've caused a deletion of a nucleotide because these two thymines were mutated by the UV radiation and created a dimer where they got stuck together, di for two. That also means that I lose one of these adenines and I am suddenly short an entire base pair than what I should have been. So that's why we want to use sunscreen. Everyone should be using sunscreen or some kind of sun protection. Wear a hat, wear long sleeves, because UV radiation can cause skin cancer in this way. We also can get mutations from certain kind of chemicals. Examples being tobacco. We know that tobacco causes cancer. Nitrates are what we use to preserve meats. Preservatives in food in general. We have a lot of weird additives and preservatives in our ultra-processed foods. Benzoyl peroxide, which is used in acne creams. We can also mutate our DNA from infections. So HPV, human papillomavirus, that's the virus that causes genital warts, but it also causes like 90% of cervical cancer. And so this virus is a concern because it mutates the cells on your cervix, which may grow uncontrollably, leading to cancer. We luckily have a vaccine for HPV now, so that's exciting. We also have Helicobacter pylori. Those bacteria can also cause mutations in the stomach lining and create a cancerous issue there too. And then we have spontaneous errors in DNA, where we just have a whoopsie, and then we end up with a new nucleotide or a deleted nucleotide or a changed nucleotide, and that can create a change all the way down your DNA sequence, possibly. Luckily, those mistakes in DNA are pretty rare. It turns out the error rate in replication is about 1 in 100,000 nucleotides, which means that there's 3,000 mistakes per replication in a human. And that sounds like a ton, but they don't stay. Because of our repair enzymes, these 3,000 mistakes are decreased. They're mostly caught, and our overall error rate in a completed piece of DNA is only 1 in 10 billion nucleotides. So our DNA replication machinery isn't perfect, but our editing ability is impeccable. And so our overall genes being altered from replication are quite rare, which is pretty nice for us as living things that do mitosis all the time. We can classify mutations based on how they affect a gene, and we have 3 main categories. So here we have our types of mutations. We're going to look at point mutations first. Point mutations change 1 single nucleotide, just 1. Here's our DNA sequence. In a no mutation example, we have TTC. When we make mRNA for that, we would get AAG. And when we look at that in our protein chart, we find that this codon codes for the amino acid LYS. Here's a visual of it. Now, in a silent mutation, I change 1 nucleotide. That changes the mRNA, but this codon still codes for the same amino acid. So I've altered my DNA, but it has no effect because the outcome is the same. It is silent. In a missense mutation, I'm going to alter my 2nd nucleotide now from the original. This missense mutation will change my mRNA, which will change the amino acid. Now that could make a huge effect. Like if we look down here at what changed our amino acid, the end of this is totally different. That may interact with the molecules around it more aggressively or less aggressively, and it might change the overall 3D shape of our protein and make it more or less efficient. So that is a missense mutation because it changed the amino acid. In a nonsense mutation, you alter 1 nucleotide, it changes your codon to a stop codon. And because it's a stop codon, you just break that protein right there. If you had a protein that was supposed to be 500 amino acids long and you inserted this in place 100, you're going to lose the majority of your protein, and it essentially makes it non-functional. So this is almost always a detrimental mutation, a nonsense mutation. We can also classify mutations based on how they're formed. So a point mutation, as we said, is the change of a single nucleic acid. Then we have duplications. I'm showing these in chromosomes, but they could just be pieces of DNA. It doesn't have to be on a chromosome. So we have mutations in random pieces of DNA, in genes, and then in chromosomes, but it's easier to visualize it like this. So in a duplication, you take 1 part of your DNA and it gets replicated when it shouldn't be. In a deletion, you take 1 part of your DNA and it just kind of disappears. In inversion, you take your DNA and nothing leaves, but it swaps places. And so you have DNA in places where it was not originally located. A translocation, this one's really cool. When you take a piece of DNA and you literally move it from one area to the next, so it hasn't left, but it's moved. It's not inverted, the order is not wrong, but it has moved to a new location. And then the most detrimental is called frame shift. So we know that codons are 3 nucleotides that code for an amino acid. I like to think about frame shift using words in the English language because it helps me visualize it. So here is a sentence made from 3-letter words. The 3-letter words represent a codon. The fat cat ate the rat. There's my sentence. Now, we refer to the pieces of DNA used to make a protein as a gene. And in a gene, we have areas referred to as the reading frame. It's the part that's going to be used. You read the codons in the reading frame. So here's my reading frame. In this reading frame, I've got 6 codons, each one coding for an amino acid. With me? Good. So now I'm going to take that sentence I made mutation, haha. Now imagine that I did an insertion into the second word. I'm going to add an extra A, and instead of deleting something, I'm going to keep that extra A, and it's going to push everything in my reading frame down. So my reading frame was pushed down because of this addition of the extra A. I can see that the rest of the sentence is still there. The fat cat ate the rat, but now it's no longer lined up. Now my codons, each one of them is changed. And so a reading frame or a frame shift mutation alters the reading frame from the mutation downward. So we also refer to this as a mutation that affects everything downstream. And that's why frame shift mutations can be so detrimental, is this single point mutation. It was an addition, an insertion, created a reading frame mutation, a frame shift mutation, and it altered every codon past it. So the following information is not gone, but it's no longer usable because the codons are shifted. Here's another visual of a reading frame mutation that's showing the amino acids. Up here, I have a normal, unaltered piece of DNA. Here's the reading frame. This is the part of the DNA that I'm using to make amino acids. I have my DNA strand here. I've got my coding strand and my non-coding strand, that's also referred to as the template strand. The coding strand is going to look like the mRNA sequence, but you use the template strand to make it. So here, my mRNA that is going to match the piece of DNA I'm using to make the amino acid is the template strand. A pairs with T, U pairs with A, C and G, T and A, G and C. So we could keep going. Each of these is a complementary base pair to the template strand. I know the wording is a little weird. The template strand is the template for the mRNA. And if we were to look at this, it looks really similar to the other side, and that's why this is referred to as the coding strand. The coding strand and the mRNA are essentially the same, except for the mRNA has U's instead of T's, but it matches the template strand. So just to get that vocab straight. Now, when we look at the mRNA, and we have our codons. Each of these codons codes for an amino acid. We can see the amino acid above it. And this is the sequence that we should get with our unmutated DNA. Now, down here, we're going to introduce a frame shift mutation. We have our DNA. We're going to insert a base pair. This is our insertion. As we insert that base pair, we've changed this codon here. And we've changed the amino acid that it used to code for. So where we used to have GLN, now we have HIS. Well, because we inserted that C, everything that was following it, AUG, AUG, gets pushed down. And now this is a codon instead of this being a codon. So that creates a new amino acid in the reading frame for everything downstream. Hence, it's a frame shift because the reading frame is altered downstream from the mutation. And even worse, this should have kept going, but our frame shift mutation introduced a stop codon, which we know is a nonsense mutation. And that stop codon prematurely truncated this protein. And so now it's inaccurate and it's too short. We also have mutations that can occur with chromosomes. During meiosis, when your homologous chromosomes are lined up on the chromosome plate in meiosis 1, if you do not have an appropriate attachment of the spindle apparatus to your homologous chromosomes, they can be pulled into separate cells, and you can end up with an egg or a sperm that has too many or too few chromosomes. We call that aneuploidy, and we have a couple common types. We have trisomy. Trisomy, tri for 3, is where you have 3 copies of a chromosome. This can really only occur in chromosome 21, 13, and 18. If it occurs in other chromosomes, one of the other 23, it tends to be fatal before birth. And even with the 3 that it can occur in, trisomy 21, which we refer to as Down syndrome, is really the only one where humans have a regular life. Like they're delayed and there's differences, but they can live for 60 years and have wonderful, happy, healthy lives. The other 2, there are less of these that are born, and the life expectancy is really short, 12 to 15 days. So we don't do well with 3 copies of chromosomes unless it's 21. That's like the only one that we can really roll with as humans. If we have trisomy, that implies that we should have monosomy. That would be 1 copy of the chromosome. This can occur in more chromosomes. So we have these as viable options. And what's really interesting about monosomy is whether or not you delete the maternal or the paternal chromosome has an effect on the syndrome. So both of these are caused by 1 chromosome instead of having 2 sets. And they are completely different syndromes with different outcomes. So it's interesting that the chromosome being maternal or paternal has an effect on the disorder. We can have a lot of different aneuploidy in our sex chromosomes. Our sex chromosomes can have tetrasomy or pentasomy. You can have 5 X chromosomes, 4 X chromosomes and a Y chromosome, 1 X chromosome and 4 Y chromosomes. These are all possible outcomes. And the variation is somewhere between common to rare. So we generally don't talk about the variation in chromosomal sex enough in biology. We like to pretend that it's a binary, and it is not a binary. Binaries rarely exist in nature. There is always variation. There's always scales of gray between black and white. Some of the common types of aneuploidy in sex chromosomes are having just 1 X chromosome, so a monosomy of the X chromosome, which is called Turner syndrome, and then a trisomy of X, which is called triple X syndrome. We also have tetrasomy for X chromosomes. And then if you are going to have a Y chromosome, you need to have at least 1 X. So if you do not have an X chromosome, that fetus is non-viable and will not gestate. But you can have X and 2Y, and you can have XXY, which is Klinefelter syndrome. And that's called Klinefelter syndrome, whether or not you have 3 X's or 2 X's and 2 Y's. So there's a ton of variation, and each one of these syndromes is different. It has a different manifestation. So really interesting and important to be aware that even when we say we're talking about chromosomal sex is highly variable. And this has nothing to do with gender. Gender is a social construct. That is a separate conversation. And if you're curious about that, you can email me and we can talk about it. It's really interesting. Humans have so many different ways to exist, and it's beautiful. It's lovely. I want to give you an example of genetic disease, cystic fibrosis. Cystic fibrosis is caused by a specific gene. And I want to use that gene to give you some practice questions right now. Here's a practice question. The cystic fibrosis gene is called CFTR. The CFTR gene is made of what subunits? Answer it yourself, and then I'll give you the answer. It's a gene, so it's made of nucleotides. There is one gene that leads to cystic fibrosis when it is mutated. How many genes total do you think scientists estimate are in the human genome? Whatever you guess is good, but the answer is about 20,000. We expect there to be 20,000 genes in the human genome. Here is chromosome 7. Chromosome 7 is where we have the gene for cystic fibrosis. These are all the disorders we've associated with chromosome 7 so far, and they're lined up in the general area that we think they're found in. So here on the long arm of the chromosome, we have cystic fibrosis. If we expect there to be 25,000 genes in the human genome, that means that there should be at least 1,000 genes per chromosome. And it's really interesting to see what we learn as we continue discovering genes. Who has a gene that causes cystic fibrosis? The answer is everyone. We all have the gene. It's whether or not the gene is mutated, leading to the disorder. Can we fix genes? Maybe. It's really cool to see the biotechnology that's coming out in the last decade. I want you to watch these two videos about CRISPR. And one of them is introduced by Jennifer Doudna, who is our scientist spotlight of the day. Her and her colleague, Emmanuelle Charpentier, she's a French scientist. They both won the joint Nobel Prize for Chemistry in 2020. So this is recent, super exciting. It was also the first time that two women had won the prize together. They both have wonderful quotes about the experience. Emmanuelle Charpentier says, "My wish is that this will provide a positive message to the young girls who would like to follow the path in science and show them that women in science can also have an impact through research that they're performing." And then Jennifer Doudna says, "I had been told that girls don't do science, and unfortunately I ignored that," which is just such a fun quote. There are some great books written by Jennifer Doudna and about Jennifer Doudna. I'm in a biology book club, and we read The Code Breaker recently. It was wonderful. Highly recommend it. So check it out. Please read these wonderful books if you want to and have time. But it's really a great insight into how CRISPR was created and the international collaboration. Like, it was literally a worldwide effort and worldwide competition. Like, there's some gossip in these books. Fabulous. Just wonderful stuff. I also want to talk about how CRISPR is affecting people currently. So this is Victoria Gray. Victoria Gray should be a household name. She's an American hero. She is the first person to have CRISPR edit cells in her body to help her with her sickle cell anemia. She is so brave. She is a wonderful spokesman for the impact of this disease on the people it affects. So just a rock star, like, so cool. I want you to listen to this NPR short article. You can read it too, but if you want, you can listen to it. And she talks about her experience with it. I'm going to have you read more articles about Victoria Gray in the lab as well, the lab that focuses on CRISPR, because she is such a rock star. And this story is amazing and needs to be talked about more. I have one about what's happening now with gene therapy. So why is it so slow to roll out after the success of Victoria Gray? Really interesting. And then read these two headlines real quick. 2018, scientists edited a couple of babies using CRISPR. It was the first time we've done gene editing in embryos that turned into humans. So it's twin baby girls. They're healthy, they're happy. He edited their genes to make them resistant to HIV, which is pretty fascinating. And a year later, he's sentenced to 3 years in prison. The scientific community totally turned on him. It's really a fascinating story. But the thing I want you to listen to is this NPR. His baby gene editing shocked ethicists, and now he's back in the lab. He speaks about what is going on now and what his goal is now. And I love the idea of the resilience and the determination. And it's just a fascinating story. All right. Let me know if you have any questions. I'm really curious to hear what you think about the current biotechnology and what's being done with it, and how mutations affect us all. So Max says, have a good day, and I will talk to you soon. Bye.